continuing our movement down the conducting zones, heading down into the lungs. We're going from the trachea into the primary bronchi, also known as the main bronchi. Now there's the right and the left main bronchi, or right and left primary bronchi. Each one of these bronchi, the right and the left, are going to enter into the hilum of the lung. Now the hilum is simply just an indent where this tubing is entering in. Now what's interesting though is the right main bronchus is wider and shorter. So why is there any difference? The heart offsets just a little bit to the left, which causes the left main bronchus to be a little bit longer and not quite as wide. So the right is wider and shorter. Plus, you're going to end up with 3 lobes on the right, 2 lobes on the left. So you have a little more heading towards the right. Now, each one of these main bronchus, or primary bronchus, is going to split into lobar bronchi, also known as secondary bronchi. In total, there are 3 on the right and 2 on the left. So if going down to the right side, you have the main or primary bronchus going down to 3 secondary bronchi. On the left, the primary goes down to 2 secondary, or you can say the mains go down to lobars. Either way is fine, just 2 different names, same exact thing. They're called lobar because the number of lobes. 2 lobes on the left, 3 lobes on the right. Now each one of these lobar bronchus is going to split into a segmental bronchus. Segmental, or also known as tertiary, as in the 3rd division. So you have primary, secondary, tertiary, or main, lobar, segmental. And these segmental, each one goes into a segment of the lungs. And from that point, just keeps on dividing and dividing and dividing. The idea is to maximize the amount of coverage inside your lung. Get to every little spot, every little piece. So these branches, every time it branches, get smaller and smaller. What you're seeing here is the trachea up top. Trachea comes down. You're going to notice right here where it splits is known as the carina, exactly. From there, you then branch into the primary bronchi. Primary bronchi known as the main bronchi. The main bronchi are going to split into the lobar bronchi, known as the secondary bronchi. Those lobar bronchi split into the segmental bronchi, known as the tertiary bronchi. So primary, secondary, tertiary, or main, lobar, segmental. So every time it splits, so here's the main, or the primary. Then you have the lobar branching off. That's the secondary. Then you have the next one is going to be segmental, going off of that. Each branching is changing the names as you go down. And that segmental just keeps on branching and branching and branching, getting smaller and smaller. Eventually, those bronchi become bronchioles. Now, the bronchioles are the smaller tubes more towards the end of the conducting pathway. Because these bronchioles get smaller and smaller, they have some structural differences. In this case, cartilage, it's absent from the bronchioles. Instead, the cartilage is giving way to more plate-like structures. Those plates give the rigidity and the structure necessary for the bronchioles. The epithelium is also changing. What's happening is that pseudostratified columnar epithelium is changing now to cuboidal. So simple cuboidal epithelium, which means we're changing that, which ends up resulting in fewer cilia and fewer goblets, almost until there's none. Well, didn't we need those goblet cells to make mucus? Yes. But as you get smaller and smaller and smaller, you have to make sure that you're not having too much mucus to clog the bronchiole. And the relative amount of smooth muscle is increasing. Now, I say relative because the amount of smooth muscle for the size of the bronchiole compared to the amount of smooth muscle in the size of the bronchi. Bronchi are much larger, so it's not an absolute, oh, look, there's this many fibers. It's just how much overall is there? Bronchioles have more in comparison to the bronchi. All right, at the end of the bronchioles, we end up finally getting into the respiratory zone. Remember, the respiratory zone is where gas exchange is occurring. That's kind of important because that's where you exchange your oxygen and your carbon dioxide. You only have, give or take, about 300 million alveoli down in the lungs volume. 300 million. Now the alveoli, that's the actual site of gas exchange. That's that little tiny sphere at the end of the whole process. And I'll show you an image in a second of what it looks like. But those alveoli are covered with capillaries. The capillaries are blood. That's where the site of exchange is occurring. Carbon dioxide from the blood goes back into the lung. Oxygen from the lung, the alveoli, goes into the capillary, into the blood. So what we're seeing is each individual one, here's one, there's one, this circle, each one is an alveoli. Together, they're referred to as an alveolar sac. So a cluster of alveoli is referred to as an alveolar sac. Now, those alveoli are going to alveolar ducts. Those ducts come up into the respiratory bronchioles. So everything distal to that, so from the respiratory bronchioles to the alveolar duct down to the alveoli, that's all respiratory zone. Moving further up, this terminal bronchiole, that's the end of the conducting zone. So the terminal bronchiole doesn't have any gas exchange. But then you go to the respiratory bronchiole down here. That's the first point of gas exchange, the first point of the respiratory zone. Everything beyond that, the alveolar duct, the alveolar sac, the alveoli itself, that's all parts of the respiratory zone. And notice over here, you're going to have all those capillaries around every individual alveoli. They're forming these clusters. You can see how it's red and blue. Well, it's oxygen rich and oxygen poor. So you have plenty of blood flow coming through here and picking up plenty of oxygen, dropping off plenty of carbon dioxide. Now, the actual membrane of the alveoli is about 0.5 micrometers thick. We're talking very, very, very thin at this point. The reason it's so thin is to optimize gas exchange of both oxygen and carbon dioxide. Now, the alveolar walls, you're going to have these type 1 cells. The type 1 cells are going to be squamose, so simple squamose, nice and flat, nice and thin. But scattered throughout these type 1 are type 2 cells. Now, type 2 cells are cuboidal. The reason these are important is they're secreting surfactant. Surfactant is going to help reduce water surface tension. Because these alveoli are so small, one single droplet of water could collapse the whole thing. But the surfactant reduces the tension and allows the alveoli to stay open. You also have antimicrobial proteins found here as well. Again, trying to keep everything healthy. Because all that air you inhaled, while you already went by the lymphatic tissue, you went by the mucus, and that traps a lot of things. Some things can still get down into the lungs and cause problems. So you're trying to make sure you still have protective measures, even all the way down deep in your lungs like this. So here's what we're seeing. This is one single alveolus. What you're noticing is all the purple, that's all the type 1 cells. But then occasionally you have these scattered macrophages in here. You're also going to have the scattered alveolar type 2 cells. The type 2 are the ones that secrete the surfactants. A little different view, a little more blown up view here. Again, same thing. Plenty of type 1s, and those scattered type 2 cells. Here's one, there's one. But the goal here, look at how thin this membrane is. The wall of the capillary and the wall of the alveolus. They're both wafer thin, allowing for optimal gas exchange. Oxygen leaving the lung, going into the blood. Carbon dioxide leaving the blood, coming to the lungs, so you can exhale out the excess carbon dioxide. So a lot of structural buildup all the way down to the alveoli at this point, from the conducting zone finally into the respiratory zone.